So today we start with interactive analysis of single cell data, but as interactive analysis might be performed with already prepared data set, and we already have to understand which kind of metrics we would apply for different data set, for different pipelines, and actually what is the variety of these pipelines. I would like to make this recap on general single cell analysis pipeline. But let's make a very, very big step back and say once more, why do we need single cell? So a lot of, it's the picture from the paper which describes different tumor and tumor heterogeneity, and not just tumor, but also other complex tissues have diverse cell types. And by using classical or bulk-based approaches, discovering cellular resolution and these changes on the cellular level was not possible before. But with the advent of single cell technologies, you already can zoom in into the single cell and see what's going on in one particular cell. So,
speaking of which, may I interrupt you for a second here?
Sure.
So, at least in some fields, in some, in neuroscience, in gastroenterology research, we have a lot of bulk sequencing data, and we have very quite a few. I would say single cell sequencing data with some specific cell enrichment or something. Does it mean that we actually cannot use bulk seq at all for our single cell analysis? Are there any tools to, I don't know, uncover this bulk seq state?
Yeah, it was a very intriguing and very popular question when people in a lot of consortia have bulk seq and would like to impute or assume which kind of single cell data could be there. So actually there is already developed set of methods called bulk sequencing data deconvolution, when you try to apply different computational tools. In particular, if you would like to analyze one bulk sample, which was already analyzed on the single cell level by other experiment, or you already have this data set, you can match and see which kind of cell proportion with a similar expression of marker genes cell profiles could be in this bulk data. However, you can also do it unsupervisedly and try to deconvolute these cell expression profiles and the cell content using marker gene. So if you have a lot of expression of one marker gene in for particular cell type, and you are tracking that in bulk, you may correlate this amount of expression and look how many cells with this marker genes you may have. Of course, it's far not that precise as people would like, but still there is a very rough way to approximate. So yeah, a lot of it's still a challenge how to match or how to make this bulk sequencing data still suitable included into these studies. And
that's awesome.
Just a very small mark, talking about single cell, the statistical significance of single cell data is lower than bulk. So actually bulk data is still very useful, but just because of this restriction and resolution, it's not so useful, could not be so used for single cell studies.
So just for those folks who investigate something very narrow or very specific, if you have just a few single-cell RNA-seq data sets published in the world, but you have a bunch of array expression data or bulk-seq data, it's still not everything is lost, and you can try to deconvolute it. But it's already out of the scope, but I think it's a nice remark. Thanks, Marina.
Yeah, so how to plan and what do we do to receive this awesome single cell data? So it's one of the showcase where you first of all you extract your tissue and try to make the cell suspension, to literally convert your solid tissue into a single cell state physically. So first of all, you do these wet lab procedures linked with further sorting or cell encapsulation in droplets. And then your main idea is to have this literally your sample in the single cell state. On this other step, on the next step, you treat your cells by lysing them and also capturing particular target molecules. So in single cell transcriptomics, it's mostly mRNA, and match it with particular barcodes which identify cells. and barcodes which identify every single molecule, and also make it useful and suitable for further procedures such as PCR and sequencing. So after this wet lab step, and the last wet lab step is a library preparation, you do sequencing using different NGS platforms. Here, actually, where the bioinformatics work begins. When you start with data processing, the first type of files that you operate with is rough sequencing files, and then you proceed them into FASTQ files most often. Other important step is to do the multiplexing, and from this whole big data set, assign which type of reads come from the one specific cell. So that is why it's called demultiplexing. And it's obvious that you do the cell this demultiplexing step when you have the cell barcodes. So sometimes you can also apply cell hashing, and while cell demultiplexing, you also analyze which kind of cell came from different batches, and which kind of cell or which kind of reads are assigned to these particular cells. And talking about after that, while you received all of this information about the reads assigned to specific cell type, you do the genome alignment and mapping this reads to genome to understand which kind of genes literally do you have, because here you might see just the sequences of different nucleotides, but you cannot impute them directly to the particular gene. So that is why you also refer to particular genome. And your first result, or first working data set, is actually this gene expression matrix or count matrix, which you then call like a raw data for single cell experiment. So for sure, these FASTQ files are raw data, but for single cell experiment, the data where is the minimum information that you can start working with is this gene expression matrix. And what you can do with gene expression matrix, and what we will discuss slightly today, is to, first of all, do this data preparation, such as normalization, filtering, scaling, then do the clustering to understand which kind of cells are there, and then some more fancy analysis. So here's a big picture. Sounds quite complicated, but literally and quite usually, all of the steps are split by different people, and that is why in group usually one person is involved in wet lab procedure, and bioinformaticians do the data analysis, and this data interpretation task is the common responsibility. So what is the count data, with this count matrix? Basically, as you can see, there is a matrix. Of course, this representation of this matrix depends on the pipeline which you use, but in general, the matrix which shows you which kind of genes, usually it's rows, and corresponds to which which kind of genes. On the first axis you have gene list, and on the other this list of cells actually. And on this table, you literally contain the information about the gene expression. So on the count matrix, you see the numbers which corresponds to the number of this mRNA transcript, which corresponds to particular genes and observed to the one cell type, one cell, sorry. However, you can also include different metadata in there. So sometimes you do not operate just with one sample. Your experiment usually is much more complicated, and that is why you have to include different metadata to perform the processing and data comparison further. So depending on pipelines, and quite often used is dropseq tools if you talk about dropseq analysis, or cell ranger if it is a 10x experiment, you received a little bit different representation for this count matrix, as I said. So here is most widely used for dropseq tools. However, for as far as I know, for cell ranger, you have just a matrix and all of this information about cells, and the genes are stored in another table. And then when you're doing this data loading, data upload, you merge this information, and as a first output, you actually make this lovely table. So what to start with, what to look at, and when you're starting doing a single cell experiment, first thing that you have to do is to do quality control check. So what do you do on the quality control check? We will see next on the example, but in general, main metrics that you take into account on the step are number of genes, so actually number of transcript that you have here. The second one is number of UMI, so actually, yeah, here, like number of transcripts, and the contaminations. So actually, contamination is a set of particular genes which you are not going to observe, or like which correspond to the damage or dead cells. So quite usually for contamination in single cell experiment, proportion of mitochondrial genes is used. Other step, or like other outcome of the single cell experiment is results is twice, sorry. So it's cell cluster. So after data processing, after different data preparation procedures such as normalization, scaling, and so on and so forth, you do clustering. And the cell cluster, where you observe the number of cells, they are differential expressed genes, like marker genes, and this interaction between clusters is one of your first output from the single cell experiment. On the next step, if you already know where to look at and your results have this actually satisfy you, you may go to advanced processing. And this step, this block includes gene ontology enrichment, discovery of RNA velocity, pseudotime, and other very interesting tools that we also will touch today. So let's start like first things first. When you have a count matrix, first thing that you do, the data upload. As I said before, you have your output from raw data processing, and literally you can just upload to the particular environment, R or Python, or you need to treat this data, like actually prepare this first run count matrix by merging with different metadata files. So first you upload everything that what you have and you would like to, everything what you would like to proceed. After that, you do quality control. Next step is normalization. What means normalization? Normalization is the step which required to solve some of the batch effects and also make your data suitable for comparison, because from different experiment, from different batch of sequencing of this like chip of sequencing, there were these expression profiles of different cells, or like of different genes from the same sample may vary, and that is why you have to actually find the proper scale, or like proper range of numbers of these expression profiles to do further analysis. So this what means normalization. If you have two variable samples, like this variability of different samples is very high, you should match this variability and still correspond the expression of different genes, for particular cells. On the next step, you do differential gene expression analysis or observation of highly variable genes. Next, scaling in dimensionality reduction. Also, quite important point, why do we do dimensionality reduction? So actually, in single cell experiment, imagine that you have 5,000 cells. And in your data, of course, it depends on these different sequencing approaches, but approximately you might have 20,000 of genes. So actually, every of these genes is a basis. So it's actually even separate dimension, and take into account and proceed all of these dimensions is quite CPU and effort consuming, and it's difficult to perform it like directly. That is why you have to reduce the number of these genes, or like of this basis of this different group of genes that you will take to account in your further analysis. So after having different profiles, you apply different clustering algorithms and receive separated cluster. best practice is to check how good clustering was performed using visualization. Usually it's a Disney and UMAP. It's like a very rough approximation how to identify like if you have separated cell cluster or not, or if your cell cluster contains is very close to another cell cluster and with different clustering parameters, they can be grouped together or split it better. So, so as I said, you can apply visual approach to understand your cluster quality, or some statistical metrics such as Yakar index, or do some other subclustering to see the cluster, to look at the cluster stability. And you do cluster annotation using this observation of different biomarkers and genes. So let's go to the most intensely used approach, how to, where actually I think everybody starts with a single cell analysis. And I think together with Alexander, we will guide you how to do that, because it will be useful. First of all, if you have not done it, it's like a must have, like must to do, which you have to do to start with single cell. And secondly, it will be useful in both cases if you will participate or will not participate in a tutorial or like in interactive session tomorrow and the day after tomorrow.
So for you, it will be actually very beneficial to listen to this lecture today, because it will give you the basic insights, and tomorrow we'll try to repeat the steps all together.
Mm-hmm. So Alexander, what do you start with in single cell analysis?
So in the beginning, I asked the bioinformaticians from the facility if they could provide me the sample they sequenced. So then we can imagine we actually received it, and there is two options. Either they provide the very, very raw data file, which we still need to process a little bit before we start with SURAD. Or the second option, they actually provide us the data files which are actually required to the SURAD. So the first data file would be the matrix, which Marina mentioned before. The second data file would be the list of genes, which is used, and the third file would be the list of the barcodes, which were used for this specific sequencing run. And basically, this SURAD program will require you to provide the three files as an input. So, for example, you have your I don't know, the brain tumor, very, very interesting sample sequenced, and you will receive these three files as an output. And if you make 10 runs, you will receive 30 files, right? 3 files per run. And you need to provide all these files to the SURAD.
But also you can provide just one file if your metrics already have this row with a number of, with list with names of genes and call names as sample IDs. And quite often you already assign the metadata like gender or some experimental condition to these call names, to like this name of the cell. So yeah, let's start with QC then. So first of all, after creating and uploading your count matrix, you try to count the percentage of mitochondrial genes. Mitochondrial genes is not the one practice or not the one parameter used for quality control check, but it's one of the most widely used. So the recommendation is from CERAP is to make a cutoff if your data set has more than 5% of mitochondrial genes. However, from the biological side, the percentage of mitochondrial genes, even for well-prepared samples, even for healthy samples, may vary and be bigger than 5%. And usually, it's better to set this cutoff not strictly 5%, but much more, to not exclude some important and informative cell data.
So Marina, just out of curiosity, when we're speaking about the filtration based on the mitochondrial content, as we previously discussed, it's very tissue specific, right? And as I understood, you are mostly working with either primary, either PBMCs, so from blood, or with neuro data sets, I would say. So could you tell which filters, which threshold do you usually use for that?
Yeah, like when I was working with brain organoids data, I used a cutoff no less than 20%. So because organoids by themselves is quite heterogeneous, and but also like I looked into this variability and distribution of mitochondrial genes to understand which cells or like which points are definitely outliers and which might not be such like bad cells or like non-informative. So yeah, but if you have perfectly set up experiment for PBMCs, you will use definitely less than 10%.
So less than 10% for PBMCs. And again, which percent usually for organoids or for neuro?
Yeah, like I remember that I used 25%, but
25, it's quite high. Yeah,
it's quite high, but because yeah, some cells in my clustering results were assigned as a dead or like cells, damaged cells. So it was quite tough for me to understand if there was literally some pieces of cells or like some contamination, or it was like so stressed cells.
So a funny aspect of this filtration step, when we have a lot of mitochondrial content in the cell, it's never very obvious whether this particular cell is just super rich for mitochondria or it's actually an agonizing cell, because all the rest of the RNA content is so low in the cell. And the thing which I didn't know in the beginning of my, let's say, single cell journey, that in the liver, there are actually very normal cells, sometimes with very high mitochondrial content. For example, the hepatocytes, which are located very close to the portal tract. And if you will take a look on the very old, okay, not old, but a few years old papers in single cells in liver, you can actually find that they use 20-ish% mitochondrial content filters. So nowadays, in some papers, people even use 35% if they specifically look for these hepatocytes. Just one of the examples. And the second funny stuff, if you will just apply the different filter for the old data sets, which are published a few years ago, but never actually studied with this filter, you can find new cell types or new interesting things about your cells, which were filtered out during the analysis. So this is a very good example why you should know and love your tissue which you work with.
Yeah, and that is why other metrics such as a number of RNA feature RNA or count RNA also apply to discover.
Sorry, very nice, interesting point. Carolina writes in the chat. So they use 20% mitochondrial content for human lung epithelial cell from fresh organoids. So that's an amazing example how different it can be. Yeah.
Yeah, that is why there is no standard cutoff. So it's very context dependent. So yeah. Actually, you can also apply this cutoff for ribosomal genes. It's not so widely used, but as an alternative for some cross-validation or as an auxiliary metric, you can use that. However, other metrics, other parameters are also used for data filtering. So when you will visualize your distribution or like percentage of your like correlation between the number of feature RNA or number of count RNA, you already might see that there is a very separated or like far standard outliers that you might filter out from your data set. So here, if you operate with a data set, as I remember, like 3,000 of cells, most of cells are grouped here, and just some of cells which we assumed are a contamination are here. So that is why you use a visualization to make the cutoff and see if you were actually to set the cutoff. But it's better to set the cutoff like not sharply here, because the most dense point, but still with including some extra data which also might be contamination.
And we have a small question regarding specifically this point. Could you please compare good quality and bad quality data? So it's something, if I remember correctly, we will receive tomorrow when we will work through the workshop, because one data set we will have super noisy cells and the second data set will be super clean. So you will be able to see it tomorrow on the tomorrow session.
Yeah. So let's move further to normalizing the data and step of data normalization. So as I said, you do this in order to be able to compare the different transcripts, or like literally the transcripts which came from different sequencing runs. Approaches for normalization, how to say it, they're usually quite diverse, but now the most widely used is this actually functions set in Seurat. So please feel free to look deeper into this technical sides.
Which normalization would you say you use the most? Is it log normalized or SC transform?
I do CT transform because CT transform is both normalization and scaling, as I remember. That is why it's more advanced. So I mean, anyway, when it was Serrat version 3, I did normalization and scaling in like 2 commands consequently, and literally did not change it so much. So actually, the merging of these functions was expected.
So
Yeah,
it's actually a very interesting point regarding normalization. There is more than one method, and with some methods, you can actually receive more biological variants to preserve more biological variants with others. I don't think we will discuss the normalization math very deeply in this course. However, we'll provide the papers regarding the SC transform, which is something worth your attention if you work with immune cells, for example, because it was specifically presented that it reveals more biological variants in these type of cells.
Yeah, so in brief, there's like in this particular case, normalization is multiplying each of these output for in 10,000 and log transformation of the obtained data. So by multiplying, you diminish this high variability and make this variability somehow comparable. And by log transformation, you reduce these like huge numbers into one scale. So I just solved the last question in the chat. So after the normalization, you are able to identify highly variable features and scale your data. So till this step, you do not operate with data so closely. Actually, you can scale data based on the subset of particular genes if you're interested in one group of genes, if you know literally where would you like to look at. But in general, usually you scale using all of the gene list and prepare your data in this way. Next step is dimensionality reduction, and as I said, in this case, every gene is particular dimension, and that is why you have to reduce this insane amount of thousands of genes into some 2,000 or even less. So usually all of these functions are already in these packages, and like if it's Serrat or ScanPy, so usually all of the steps are already included in the software which you will use. The first step of the dimensionality reduction is principal component analysis, and actually it also can provide you the understanding of the most influential factors. I mean, on the genes which influence the variability in your data set the most. And that is why you do, first of all, the principal component analysis. However, you can also apply canonical correlation analysis and other statistical approaches, which will do the same. So actually display or like find the dimensions or like components which have the most impact on your data set. Yep. So after this reducing dimensionality, you do clustering. Also, a lot of like a huge variety of clustering approaches.
May I ask you first something?
Yeah, sure.
So why do you think it's even important to make dimensionality reduction? I mean, we have this beautiful matrices with all the cells and all the genes we need. So what's the issue? Why do we do this?
Yeah, like imagine that you, first of all, you are limited by computational power, and it's difficult to handle all of this data set afterwards. So like you can actually operate with data. Secondly, just particular number of genes in this huge list varies. So actually just some set of these genes may tell you something because, okay, you find a different transcript, but quite often they do not have any changes, like any impact, like the information of them might not be so relevant in your experiment. That is why you do dimensionality reduction as well.
For me, it always was very basic explanation, because if you have one cell and 20,000 genes, so you have basically 20,000 dimensions, and imagine you have, I don't know, many thousand cells actually. And for humans, it's actually quite difficult to plot something meaningful with this number of dimensions while being able to see what's the difference between these cells and these cells or something like that. So here dimensionality reduction helps us a lot. And I know a lot of very smart people working on new methods. So actually there is a lot of those. And I think which one they use in this tutorial?
You mean which one, PCA or like for
Yeah, so they do
dimensional?
PCA and also they do UMAP, right? Yeah, I think that's okay. We'll go closer to that point later.
Yeah. Well, can we get the link from the website? Yes. So it's like Sarah tutorial, and I will post the presentation today in chat. So, but feel free. And actually, I already seen that one person shared this link in single cell day 4, our channel. So feel free to find it. So when you have all of this prepared data set, you do clustering, and there are multiple approaches of to do clustering. You can do like Leuvian clustering or key nearest neighborhood, but in general, you can also apply every other tool which you like. It's just something that you have to be sure that it will give you some statistically significant and informative results. And also do not forget to specify it in your publication with this material and method section. Yes, so after clustering, you can do already non-linear dimensional reduction, because previously you did linear dimensionality reduction, then you apply different tools, other set of tools, and visualize the results. So here, after processing your data set, you can see the different cell clusters, and what you do not know is actually what kind of these clusters. You know that, like, for example, something greenish is staying far from other cell types, and some clusters from 4 to 7, as I see, are grouped into one cluster. So actually, it's not so informative, and that is why you have to do downstream analysis and identify this cluster. Alexander, what do you do for cluster identification?
Oh, actually, that's one of my favorite points, because when you receive your precious data set that you received after, I don't know, many weeks of establishing the protocol, and you see this number 1, 2, 3, 4, 5, you obviously want something more relevant for you, right? So there are guided. Oh, actually, there's a lot of different methods. So what I usually do. Number one, I know which markers my cells usually express. So for example, if I study hepatocytes, I know that albumin and some specific receptors, specific channels are specific for these cells. So I can just take a look which clusters do express these markers. Usually it will be a few clusters. In this case, I will say, okay, so this is a hepatocytes cluster number 1, number 2, number 3. Another, and the same goes for any markers I would specifically want to measure, or if I just care only about these markers. The second approach, which I usually use if I work with new data set, I just take a look which gene expression markers are differentially expressed for this specific cluster versus all the rest of the clusters. And then, for example, I can see that, oh, it seems that cluster number 0 expresses very high number of albumin gene or some specific receptor, and then two options. Either I know these markers and I just immediately write, okay, hepatocytes, or I just go to the public database and see which cells usually express these markers. So as you can see, it's a lot of approaches, and it really depends on what you're looking for. If you want to just review all the new cell populations, most likely the second approach, which is just taking a look on the expression markers, will be more beneficial for you. If you're mostly interested only on your cells, if you make some specific enrichment experiment and you know what you're looking at, probably just measure your markers, which you already know, and take a look.
You say that you look if some particular gene is expressed in one cluster and not expressed in other cluster. How do you do that? Like, how do you estimate this difference or like significance of expression?
Well, it's always a question about how much is marker expressed. I mean, there could be a chance that, for example, specific interleukin gene is expressed in one cell cluster very highly and very moderately in other cell populations. So I usually try to, well, when it's possible, try to evaluate, I don't know, top 10 or 20 most significant genes per cluster and see if I don't know what I'm working with. So that's what I usually do, just not to miss anything significant.
Yeah, and to talk about the metrics, because manually picking genes and inserting into different tables related to, like columns related to cell clusters is quite time-consuming process. So you look into different statistical metrics, and by working with your data set, you on this linear or non-linear dimensionality reduction step generate some new data representation with already normalized, scaled, and processed data. So, and how to understand if one gene is expressed in this particular cluster. So, first of all, you have to identify and assign this. Okay, you have to a little bit process this table and see literally clusters of what correspond to different cell types, and assign and look into two values, which actually I look at. So the first one is average log to FC, and the second one is p-value. Why do we look at p-value? To understand the statistical significance, or like the confidence which which is correlated to one particular gene. So you can see that this gene, this confidence that this gene's expression is presented in this data set is pretty high, because the p-value is extremely low. But answering or like supplying Alexander's approach, how to understand if your gene is expressed in your cluster, in one cluster, and not expressed in other, this parameter shows you these differences in the gene expression. And while you are looking into this data, finding the markers of particular clusters, you should analyze, and it's even easier to order the data and find these markers in the table.
There's actually a few very interesting comments regarding the adjusted p-value versus usual p-value considering the sample size. And yes, I think you're absolutely right. So adjusted p-value, that's why it's already included in the standard output of such functions, because it's really, I would say, more valuable for us here. Yes.
Yeah, I agree. Like,
but also, of course,
because they correlate, I was like picking the incorrect p-value.
What is also important, when you compare the expression of different clusters, you are actually free to select the statistical test you would like. So I'm not sure if we will have time to do it tomorrow, but there is, I will just send it to the Slack. In the description of the function, there is a list of the statistical tests you can actually apply to this specific differential expression analysis you're making. So if you know that, for example, you would like to receive the specific output, you most likely have an option to perform this specific test. So I will just send the link to Slack.
Yeah, so other steps are related to understanding and like making us sure that this particular gene is actually a biomarker of the cell type. And you can apply different visualization to do that. For example, here are the violin plot that shows different distribution of expression of different genes in these cell types. So here, this identity is 9 cell types, like Y9, because 0 cell cluster is also one cluster of cell, like cell type, and this expression level. So here you can see that these genes are cell type specific markers, because in biology, it's also possible that one marker is shared by different cell types, or actually it's not a cell type marker, but process marker. For example, quite usually on your representation or your visualization, you see even the cluster of dividing cells, because they have this cell cycle genes intensively expressed. But also you might plot this top to A or like the other cell cycle related genes, and you will have them in different cell clusters. So you use, in brief, you use this visualization to support your hypothesis about this particular biomarkers and so on and so forth. Like here, different, the one biomarker is presented in different cell types, but the amount of expression is so different, like so variable. However, you can also plot this and see the distribution of these biomarkers on the UMAP, or like t-SNE, which you use. Like, we will talk about UMAP because t-SNE is quite old schoolish way of visualizing and processing like this nonlinear dimensionality reduction method. So what you can do is you can highlight the cells which have these genes. So you insert the vector of genes in your data set and understand if this gene is cell type specific or not. So it's in brief. The last and quite widely used visualization related to clustering is this heat map, which shows you which list here are assigned to different cell types, and by comparing these colors, so like super yellowish is a high level of expression and deep purple is this low level of expression, like by this clustering, you might see how these particular genes are cell type specific. It's very informative. For example, when I was lazy to look into different tables of this expression, I just downloaded the heat map and tried to do this cell type annotation. Because, for example, I plot top 5, top 10, top 20 genes from my data set. And they are assigned to a particular, not assigned, like in here on this heat map, I see their expression in my cell clusters. And then I can see if I can pick one particular gene, see if it's a marker gene, and see, look at that expression here and assign it to the cluster one. So if I know it's like, this is I don't know, leukocyte monocyte marker, then I picked a particular cluster.
There's also a question regarding the heat map. So there is also an interactive way to access heat maps, right? And actually many of these steps. Is it correct?
Could you please repeat? I did not.
So there are also interactive ways to access this many of the steps, right? The shiny apps.
Yeah, like we will talk about that. Like
okay.
It was my decision that I started working on interactive data analysis too, and I realized that we cannot guide our participants by like literally into single cell processing. That is why we are finishing with Serat and we'll go to interactive data.
Perfect. Then your question will be answered slightly later.
Yeah, yeah. So lastly, you assign this to the particular clusters and save your object with already assigned cell type identity and also different like process data on your PC. So literally do not forget to do that because it will be pity to lose so much time and power. So the question that I see on the chat is like, what does UMAP1 and UMAP2 mean? It's like a dimension which you receive by this nonlinear dimensionality reduction, and actually it's two axes where your data is presented. So similar to principal component, it's this axis where you have this 2D representation, because do not forget is you have this like extremely highly sparse data and with many of dimension, and you would like to look at them just on like two-dimensional space or like three-dimensional space if you have these 3D UMaps. So yeah, here already the answer.
Importantly, it doesn't mean any human readable. It's like it's not movement versus time or something. So it's basically just basically plots the dimensions against each other. So in this case, and you can see how that cluster.
Yeah. So let's look into different examples. So I used several articles to highlight which kind of which visualization and which results did they get with single cell data. So in this study, they operated with as you can see, like ice, and they extracted different cell types and described their features for the experiment. So here you already can recognize the UMAP representation, and here the features plot which describes the different level of expression of the marker genes. So I just would like to speed up because I would like to be in time with the end of the session. So here the cell type identity was assigned, like matched with different violin plots. So they here on this graph below, they show different cell types, which you can pick from there. And here, these cell type specific markers, which they use for cell type identification. other studies related to fetal ovary development use the same 10x, use the 10x protocol and the same procedures. And here already it's easier to recognize these UMAPs because high, like with this high confidence, I can say that they were done in Seurat. But also on the plot nearby, I would like to show that on this UMAP, you can plot not just cell types, but also the origin of the different cells and samples in your data set. So basically here are these data received from different samples, in particular ovary on the different stages of development. And that is why you already can see which of the cell types, cell clusters are assigned on different cell samples. So, and on this step, you can also decide what is better to do. Maybe it will be better to split this data set completely before the data processing. Please do not write, guys. No, I will not allow you to do how to do this. Okay, I disable annotation. Yes. It's quite confusing if you write on the screen. You can do this, not just cell type plotting, but plotting other metadata, including your samples. I believe that it's very relevant. Oh, now I am annotate. No. Okay, and the feature plot describing different cell type specific marker genes. And heat map, so it's like the classic of Seurat tutorial. However, let's talk about other advanced analysis, which you can do with single cell processing. In both, you can do this in manually, like in R or Python, or in different interactive tools. So cluster observation is widely used in interactive tools, but these few approaches like differential expression, pseudotime, I'm not sure that I've seen some already open access free software which allows you to do that. Like BioTuring browser, which is commercially available, allows you to do that, but open access tool not. But let's talk about differential gene expression. So here you can also see these marker genes and why and visualize them by different using different metrics. So you look into differentially expressed genes in one particular sample and can recognize genes which are highly or low expressed comparing to other cell types. So actually marker genes like abundance of one transcript is a biomarker, but as well as absence of one of particular mRNAs are also the biomarker. So to visualize pseudotime, and what is the pseudotime? Pseudotime is the process when you observe your data in the perspective where you try to look at the development process in your sample. So for example, you're working with a highly dividable cells, so with some progenitors or maybe IPCs, like as we already see from chat from participants, some organoids data. You might also have a lot of undivided cells or like progenitors, which will be like even on the snapshot, which you can also see that these cells form other cell types. So by using different pseudotime, you align your object on your data into different developmental trajectories. And how do you visualize this? This information on gene-based level. So here you can group them by KEGG enrichment, like assign particular gene sets to the processes and see how like the gradient of their changes. So that is the approach how to visualize the pseudotime, and it's mostly relevant to understand the disease projection, like disease or like developmental development process. Development. I mean. Projection. Yeah, okay, so the developmental process in biological system. Yes, and as was used in pseudotime already, like enrichment on gene ontology terms. So actually, what does it do? It groups the gene enrichment uses the genes of interest which you can observe in one cell type or like group of cell type, and assign to particular biological processes which were already discovered. So for example, if some of genes are the genes related to cell cycle or like heart contraction, they will be linked to this particular process. And that is why you can make your output much more informative and assume that these processes are, for example, active or like intensively developed in one cell types and decreased or not presented at all in other cell types.
I would say it's rather an approach to make biological meaning of the data you receive. Because if you're a bioinformatician, there is a big chance that after you process your data, you will have to go to the lab meeting and to meet the biologists and to explain, okay, so in these cells, we have these pathways or these biological terms, in this case, over enriched. And in this case, they can understand you. And then when you have something interesting, for example, I don't know, cytokine mediated signaling in your microglial cells in brain, then you can go deeper and actually see which genes are overexpressed. Because if you just send them a big CSV file with I don't know 500 of genes, it can be a little bit scary, and this is much more human readable.
Yeah, and as well as plotting different genes, you can also plot this gene enrichment terms. So yep. So in brief, when you do processing of single cell experiment data, you have output and understanding of the quality of your data received by QC control, so the very early stage. After that, you have list of differentially expressed genes in this list of genes assigned to particular clusters, and the clusters with their features. So the number of transcript and like size of the cells related to clusters and so on and so forth. So output and like input and output as well, which you processed in your data set, is CRAT object if you use CRAT package, like a single cell experiment object and loom object. So actually, I saw the question first of all about uploading different data types or like data tables from available databases, and usually it's very rare that they will be already in CRAT or other other types of single cell objects. However, you might upload them as a TXT or a CSV file. So it's also some kind of output that you can use, but it's much more convenient to use these complex objects. It's usually S4 objects, which already include a lot of like blocks with metadata and so on and so forth. So to handle different kind of data set and to be able to proceed data set from different pipelines, you have these objects. And last comment from my side, like loom object you use if you work with very big data sets. So it's like just some technical optimization of data set processing approach. So we have less than 5 minutes, but I will be try to make it fast to discuss different web tools and databases where you can do the single cell analysis without coding. So there are already a lot of initiatives which, first of all, support and make the some kind of basis for collaboration for different scientific group who works in single cell analysis. So the one, the most famous is Human Cell Atlas. Yeah, and however, all of this, as well as Mouse Cell Atlas, but this list of these hubs and these databases increased. So the one positive moment, so like what you have to know about these databases is that these tools have, like these resources have information about the publication, about the data set, about like related information like metadata and the result that was received using after processing of this data set. So it's actually very convenient. But some advanced options may allow you to do also the discovery of your single cell objects or already published single cell objects using web interface. So here is a single cell expression atlas. And what you can do, and you can try to browse different genes of interest, and then you will see in which experiment you can find them. So it's actually, if you are working with one particular gene and in one, like in one species, like, yeah, so you can observe this deeper and see the most relevant data set for yourself. But also, you can go to a data set and interact with them using this resource. So here you can see different kingdoms of nature, and you can find your organism of interest to work with. So here are different studies. Let's look at that. And what do you have? As I say, the information about the publication and actually the visualization of this data set. And you can look deeper into into metadata there and understand this topology of clustering output using this resource. So also you can download the data from here, but I think the biggest advantage of this tool is that you can browse the different single cell data. That's what I like the most. However, single cell expression atlas is not the one tool. Also, you can use this cell browser from UCSC. And I like this tool much more. I have been working with it quite a lot because, as I said before, I was working with human brain organoids, and they already have this data set or like reference data for these kind of studies. This awesome tool summarize the information about different meta-analysis or separated data, like independent data set. And for example, like it's the first time when I open it, I hope it will be fast. So, and what you can get from here is also the output of clustering. But what I love more is that you can also see this deeper cluster annotation to see, look in this cluster expression matrix already with normalized data. So here you already can recognize different metrics, which you will also see in your single cell object, and look into the different genes which are here. Also, what is very comfortable or useful that you can visualize different metadata or like particular cell types from here. Yeah, so I recommend you to use this meta-analysis because imagine you have quite a widely observed object with a lot of data there, and you might also be interested in this meta-analysis. So making meta-analysis in this data set and this ability to operate and interact with this data set is quite effort-consuming and quite complicated task. So that is why it's easier when you already have some kind of meta-analysis output here. So UCSC cell browser, feel free to find your lovely data set in this published list. And keep track because probably soon your object of interest will be published here. Last tool, interactive tool that we will touch is Single Cell from Broad Institute, and it actually does the same with a Single Cell Experiment browser from Amble, as I remember. So feel free to look into the functionalities of this data because usually the downloading process is quite intense and takes some time. And that is why I think that it will be some lovely homework for you to understand which tool you already might use and what results or like which insights you can get from published data set and such tools where you interact with this data set. So here again, from single cell portal, you operate with different clustering. You can pick different cells, observe like here, every dot is independent cells, and look into their profiles. But it's not it's not all. There are a lot of other other tools which allows you this interactive visualization. I think for people who are already familiar with single cell, it will not be some new discovery, but one of these freshly released tool, which also allows you to analyze well-described data set called Azimuth. It's the project or like part of CIRAT, where you can analyze your data set in even single cell multi-omics perspective. So on this tool, you can match and observe the data like previously processed by the group members from the TG Lab, and track, actually match your dataset information from dataset information like presented in Azimuth. So this tool allows you to first of all do clustering, do automated cluster annotation, like do all of these procedures on single cell, and analyze, like do this automatic cluster annotation by using biomarkers and other cell type genes, cell type specific genes from studies which were already done. So actually it's very useful. And also Azimuth allows you to do imputation of site-seq data. So remember that Vladislav described on the day 2, that you can track and do sequencing of cell surface proteins. And that's why you can do this beam-omics, like multi-omics discovery, and match, actually, like impute this site-seq profile, like profile of surface proteins into your data set, also using this tool. If we will benchmark all of these tools, here you can see Single Cell Explorer and UCSC Cell Browser and other tools which are not web-based, but you can integrate and use them as a Shiny app or like some software, also open access. They are pretty the same, but still some overlap on like on functionalities is missing. So most of these features and like these advantages of this tool include this selection of cells. Like remember when I was picking one particular region of cell of interest in the data set, like zooming in, zooming out. Also, it's like just some user-friendly interface, which also can help you in single cell data exploration, like embedding of data, like gene expression plotting, as we discovered in UCSC browser, differential expression, and so on and so forth. I set the link of this benchmarking paper here, and please feel free to check it because it's open access, so I think it will be useful spending of time. And here is more advanced comparison of these tools. So as you can see, some of them are web-based, some of them can be used in Docker container, and some of them are commercially available as a software as a service. Here you can see the differences on different types of input files that you can upload them in as a single cell experiment. So as we discovered this Loom object, which handles huge data sets, CIRAT, single cell experiment, or like regular tables. And they're realized on different platforms, and also some technical details on the processing of this data set, how much it will take, and so on and so forth. So there are already a lot of tools developed, but still they have some restrictions. And which kind of limitation, like, to finish with this part, which kind of limitation do interactive analysis tools have? First of all, is the size of the data set. So as you might see on the previous slide, this size of data set influence the time of uploading the data set and processing. So imagine that if it's a web-based tool, it will take insane amount of time if it's a huge data set, or will be simply rejected. Also, the functional analysis and other types of analysis are very restricted by functionalities of these tools, like comparable with code, where you can apply other data set or other libraries or other packages which were not even used for single cell, like for general deep learning or statistics. And also it's related to this manual control, how to set different parameters, apply different like clustering approaches, and so on and so forth. So using tools as a wet lab specialist is quite fancy, but if you are pretending to be bioinformatician, you definitely have to code just a little bit. Maybe I believe that after our practice on the weekend, you will be experienced with that. So here, lastly, would like to briefly mention the small GitHub page that I developed this winter. So this web page includes and will help you, will guide you in the single cell analysis, how to become more confident in this process. I mean, like where to start. I highlighted the main tutorials here, where you can use, which you can use for your study and yeah, for advancing your skills in single cell analysis. And also I collected different papers. Most of them are open access. I really looked to particular papers that everybody can use for his or her studies. So yeah, please feel free. I will be glad to, first of all, receive your feedback and also know that this is something helpful for you. And yeah, and I believe that it will help you also after this course. So I think that's all from my side. End of the first session. We are slightly late. I hope it's okay for you.
An organizational moment, I propose we'll not stretch time too much, so we'll still finish around 8. So now it's 7:07 Vienna time, and I propose we will announce that we will not begin the second part before 7:15, so you have 8 minutes. In the meanwhile, we will try to answer the questions which are already here, and if you want to, make some tea or coffee, feel free and return back at 7:15, or feel free to stay to receive the answers. So if it's okay for you, I can just read your questions, which I have recorded. And we can, yeah. So a lot of people actually asking about the GitHub link, everything. So Marina is the person who immediately shares her lecture after the event. So you will receive it on the Slack channel immediately after we finish with the sessions today. Don't worry about that. So the first question, 10x microfluidics. It means that there are small microfluidic vessels, there are small gems or these bubbles. May it happen that two cells instead of one cell will be trapped in one bubble and marked with so two cells will be marked with the unique molecular identifiers? And if yes, what can we do with that?
Yeah, like I believe that we already had this question. So first of all, unique molecular identifier will be related to RNA. So don't worry it sounds quite contradictory. You will not have, probably you might get the same cell barcode, but still it's quite often happening that you get two cells in your droplet. And actually, you can apply different, so you will filter the cells by quality control metrics, such as a number of reads or number of amaron features, and that this doublet probably will have in total more sequences and more molecules than the other cells, and that is why you might see that and filter as a doublet. So it's not, you are not sure that it's some relevant information for you. So you can use this. However, also already more advanced tools which use more complicated statistics were developed to analyze and detect these doublets. So actually, yeah, you can detect it by applying packages or playing around with QC metrics. But I recommend you to think of that from other perspective, because I was really surprised when my supervisor shared with me one paper where people literally looked into this doublet. So actually, these doublets means that, of course, it could be some technical issue that is like mixing was not correct. But actually, if you have these doublets, it means that your cells were interacting one with each other, and that is why it could be used for this cell communication, like cell how to say it? Cell interaction analysis. Because, as we said, you dilute everything to cell and do this single cell suspension. And you would like to have this in this particular droplets, but still you may also be interested in this cell interaction, and that is one of the most valid tool to do that. So yeah, be careful with doublets, and it's not so bad.
Yeah. Well, that's an actually very interesting argument regarding doublets. I have never thought about that, and That's something I'm going to think about this session. Thanks to you. The second question was from Andrea. So what about the quality control and trimming reads before mapping? Which parameters do we use for that? So for that, it's a very simple answer. It's not the SURAD, but it's something before SURAD.
Yeah.
If you use 10x Chromium to prepare the samples after you sequence, sequence your sample in Illumina platform, you'll receive the BCL file, which is the raw data. And then 10x developed their own software, which is proprietary but free, so which calls scRNA. And actually, you need to perform two very simple commands. So the first of all is scRNA make fast FASTQ, so it will convert this BCL to FASTQ file, and the second, so it will perform the multiplexing, and the second will be scRNA count, which will generate the barcodes, gene, and matrix files, which you need to use for the further secondary analysis. So there are some options or arguments you can pass to these commands, which are depending on which library preparation you used, so there are some things you need to figure out. And in the manual, they quite good describe what you should use when. So basically, these steps are performed during this scRNA step before SURAD. Yes, yeah. And the same you have to do, by the way, for the rest of the participants. There is an option to download the raw data files from the public repositories after studies. Sometimes people publish actually these 3 files, barcodes, genes, and the matrix. Sometimes they don't. And if they don't, but they publish just raw files, you have to demultiplex and convert it with the scRNA. And and the question here is about not 10x. So here I'm afraid I cannot answer because right now I'm working only with 10x.
Yeah, okay, I will try typing. It's like
so you recommend drop-seq tools, PHX tools in drops tools?
In drops, like a lot of as I remember, it's less than 10 tools, but actually already developed tools cover all of these needs.
So the next question regarding the slide number 10, when you showed the matrix, there were, y-axis were genes and x-axis were control and experiment. So the question is, are these control and experiments are all the cells in the case of single cell sequencing, and why they are denoted differently? So what can you say about that?
Why do we have the same genes for the different cells?
I just think that the example you showed in the matrix was from the bulk-seq experiment, right? Because it was just the control versus treatment. And usually we see just cell number 1, number 2. I mean, they're not named one and 2, but still it's in principle there are this cell number 1, 2, 3, and however much you sequenced, however many you sequenced. And the y-axis is always, for example, in this particular example is genes. So you always see the information for these specific cells, which genes expressed how much. That was the idea, I guess, so of that demonstration. So the question number 4, the count matrix for single cell RNA-seq is going to have data at few thousand cells with over 20,000 genes. Is it correct? Well, yes.
Yeah, like of course it depends on the number of cells that you will capture. Because probably imagine you have like sample size. How do you measure it? Of course you measure like maybe some, I don't know, you measure the cell concentration and amount of your solution when you do the self-suspension, and also you can assume how many cells do you have there. But imagine that in your cell suspension, far not every cell will be encapsulated into the droplet. So the conversion, depending on machines, is no more than 30% as far as I know. So yeah,
but if you
depends on the number of cells.
But if you submitted 1,000 of cells and received 10,000 of cells sequenced, maybe something also was wrong.
Like it's not a good result.
That's something paranormal already. Okay, so the next question would be about and I think we will be really fast about the filtering regarding to the mitochondrial content. So isn't it a bias if you discard a type of cells with higher content of mitochondria? So the brief answer is you of course can not do this, but then your data set will be super busy because there's a lot of dying cells during this procedure, right? Because they're going, on all these buffers and go to microfluidics. And when the cells are dying, they also can express different markers, for example, related to apoptosis, related to the cell death. And it's a question, if you would like to risk and include the cells to your analysis and see that, your healthy tissue expresses the cell death receptors, or maybe it's better to just filter out the cells which are most likely dying. So that's
Yeah, but also like imagine that first of all, a lot of technical drawbacks are already in single cell. So imagine that all of this by not filtering this particular cells, you will be unable to cluster them properly. So of course, it's quite okay if you're sequencing by itself and encapsulation, like before, like preliminary procedures was done very nice, and then you can filter out this dying or damaged cells from your object. But it's better to like set this just some of your of some of this cutoff. But of course, nobody restricts you to play around. So it means that you can also just set different cutoffs and see how it will be.
Right. So the question number 7, how can we know how many cells do we have in each cluster? And I think in the draft for tomorrow's workshop, I actually introduced this command. So you can make the data table when the column number 1 will be your clusters, the column number 2 will be the count of the cells per cluster. So it's something you can make relatively easy computationally. The last question, what type of data can you use for azimuth? So I guess it's regarding the input data, which what you use, because I have no experience with azimuth, unfortunately.
Yeah, I do not remember by heart that issue. I just remember that it existed. It was like, some manual procedure from my side, like for sure Serrat object, no
Okay.
doubts, but I'm just not sure if it will be like some other data types.
So the short answer is it's better to check the azimuth manual.
Yes.
Unfortunately, personally, I didn't work. I am a simple person working with Serrat in my environment for a few months, updated every month. So yeah, that's the short answer. So it's already 7:17. Should we proceed with the second part to be able to finish? And of course, during the second part, you are more than welcome to ask questions on Slack or here in Zoom, and we will try to have time for the discussion after, not to take you too long on the Friday's evening, or not evening if you're in different time
So
zone.
you might be able to see my second screen.
Yes, we see your second screen. So it will be an interactive session, right?
I hope so. Like, yeah, the idea is that I will try to present some of this data, or like try to draft with you and guide you with this notes and like some interaction in our session. So yeah, let's start with the beginning. We are talking right now about the single cell multi-omics, and in brief, why it's important and why it's so cool. So before we were talking about single cell, single omics. It means that we looked into different single cell transcriptomics, like it's single cell RNA sequencing, or single cell epigenomics, like single cell attack sequencing, or this also we touch a little bit single cell transcriptomics by single cell site-seq. So what about single cell multi-omics by itself, and why people do study it? So imagine that you have several dimensions. Oh, okay, so in brief, step back. Single cell multi-omics was awarded as a method of the year in 2019, and it's really extremely powerful, informative method. Because as you might know, processes in cell are very complex and diverse, and by studying different modalities and different changes on these molecular layers, you are able to have a more comprehensive results and insights from your data. That is why several, like multiple of different single cell multi-omics approaches were developed. However, still, there are a few main challenges of single cell single cell multi-omics data analysis, which includes this isolation, proper recording of cells, and sequencing by itself, or like this discovery of these omics modalities. So this set of challenges is related to wet lab. But also data integration challenges exist. So you might also be able to integrate all of this huge data set which you generated for your sample. But let's become closer and more familiar with different single cell omics tools. And let's make a note, or like make a graph of different single cell multi-omics methods. So let's start with this method, which already are well known for, which are linked to single cell transcriptomics. So if you will see that single cell transcriptomics could be integrated with three main different modalities. First of all, it was like the most obvious that by studying single cell by themselves and like doing sequencing, you might also go deeper into the genome sequencing. Please, guys, if you have some problems with this interactive session, or like Alexander, please help me with managing
it looks cool. People just ask you what's the name of the software. Maybe they want to try it.
Yeah, okay, it's a concept and even it's open access. So it's like concepts.
Concepts, okay.
Concepts, yes. So I really love these colors. So I really love making some notes here. So let's discover the single cell transcriptome and genome methods. So the first method that we are going to talk about, and I will just briefly mention some main features of this method, is DR-seq. DR-seq stands from DNA and RNA sequencing, and it's a semi-manual method. Uses a pipette for cell isolation there, and the preamplification and tagging of DNA and RNA. So by using this tool, you can observe different copy number variation and their copy number variation of DNA and the impact on the transcriptome profile. So it's already very informative match and insight of the data that you can look at. Another tool, so I really will try to make it fast, is GNT, or genome and transcriptome sequencing. And this method also, comparable to DR-seq, does not include manual picking, but it's automated by flow cytometry and DNA-based bit separation and polyadenylated RNA. After finishing with one block with like genome and transcriptome methods, I will show you this their like wet lab procedure underlying these methods. So let's just briefly talk about the method by themselves. So one other method is used for genome and transcriptome measurements in single cell called CIDR, and it stands from simultaneous isolation of genomic RNA or genomic DNA and total RNA. So this method allows you to do see copy number variation discovery as well, and the differences of CIDR and other tools, like it's more similar to DR, that you also do manual cell isolation and analysis of DNA and RNA using regular, like already discovered techniques. The last method, no, not the last, almost the last method in this section, called target. And or target-seq, it's a method based on the fluorescent activated cell sorting used for cell isolation. That is why it's a high throughput method. And it's based on reverse transcription and amplification of your target molecules, like in this case, reverse transcription of mRNA and amplification of both DNA and RNA, and consequent library preparation. Interesting point that target is a mutation sensitive method. So its preliminary was developed for tracking mutation in single cells. And other method, which also includes genome and transcriptome sequencing, but also allow you to do epigenomics analysis, is called TRIASEq. So I need to to do some removals. Yes, so here on this border of genome and transcriptome, and epigenome stands TRIASEq. So sequencing of three modalities. And actually what's going on there, you sequence genome, like first of all, you separate your cells using FACS, then do cell lysis, then separate your DNA and RNA using centrifuge. After that, you already split your DNA and RNA content. You do independent analysis of DNA, such as like library preparation, and RNA processing using single-cell RNA-seq. Methylation is, in this case, also used, and this methylation is measured by a single cell RRBS tool, which we will discuss just a little bit later. So what it allows and where it was firstly developed, it was developed for cancer cells and also oriented on copy number variation and methylation changes in these different cancer cell types. So here is just this small explanation of DNA and RNA simultaneous measurements in single cell, and actually which protocols are underlying on this method. So with this single cell triassic, which was the last in our talk, you do lysis of cell, and like as we say, first of all, you separate your cells using FACS, physical separation of DNA and RNA using centrifugation, lysis of nuclear content, and processing of DNA and mRNA using single cell whole genome sequencing, single cell bisulfite sequencing, which is oriented on methylome, and SMART-seq2. SMART-seq2 is a full-length RNA sequencing protocol, which has smaller throughput than DropSeq, but more in it's more informative if you are targeting one particular small set of cells. So yeah, in DR and GNT-seq tools, you also start with whole cell lysis, and before you pick the cells manually, and then you in DR-seq you apply this. You perform this reverse transcription with barcoded primers and how to say go into this non-standard amplification of this molecule. So there is one of the approach of amplification of nucleic acid called Malbec. So it's like multiple annealing and looping based amplification cycles, which allows you to prepare both DNA and RNA for cell-seq and Malbec for DNA discovery. And similar to DR, after the splitting in GNT protocol, you start with separation of DNA and RNA by using bits as well, like bits for capturing mRNA. And then you have the separated suspension of DNA, which you proceed with whole genome sequencing, and these cells like mRNA reads captured by bits with poly-edited tails. CIDR includes different antibodies conjugated to magnetic microbits, and by targeting these cells, you by capturing this, by processing this in such way, you have the cell lysate and also able to separate different DNA and RNA. So by processing by implementing these magnetic bits, you capture RNAs, and like similarly to GT-seq, but you also use bits for separation DNA, RNA, and then you perform separated treatment for DNA, for whole genome sequencing, and single cell RNA. And the last method is target, when you treat your cells with proteases in order to digest all of the cell content and do this particular processing. Okay, for target, I have a separated slide. No, sorry. Oof. I thought, I think it's just like, I was sure that I have it. Okay, so why do you do that? You also have to target your mRNA with this particular sequences in order to separate and mark the DNA and RNA for reverse transcription and sequencing by itself. So you do this enzyme treatment to how to say it, split, not to separate this DNA in mRNA reads, which you will see in the libraries, like you mark by these enzymes mRNAs with particular primers, and then do literally regular procedures of reverse transcription and then preparation for sequencing. So yeah. What about epigenome and transcriptome? Also, some fancy methods are already developed, and I will try to make it good looking. And to one of the first methods which was developed, called single cell methylome and transcriptome-seq, so MNT. And this method started with manual cell isolation. And first of all, you lyse yourself, then you separate your genome DNA and mRNA by magnetic bits, and then use this genome DNA for single cell whole genome bisulfite sequencing. Yeah, and RNA for processing, like for classic single cell RNA sequencing. So what is good and like what is useful in this method is that non-CpG islands are promoter methylation and transcription are anti-correlated. So actually that is why you can look into the epigenomic regulation performed by methylation and changes in different transcripts. And that is why you can link this genetic like regulatory construct and the output of their activity in your data set. Similarly, by name, please be careful with that. There is a method called a single cell MT sequencing. So the difference is just in this ampersand. And this method is a little bit more throughput, and it's based on flow cytometry for cell isolation. Then you do, again, bead-based separation of DNA and polyadenylated mRNA. And then you lysate, like by this separation, you already can treat your DNA for whole genome bisulfite sequencing, here is different, for RSB, yeah, RBS, I'm really sorry. Sequencing for tracking the methylome and transcriptome using regular single cell RNA-seq. So what is single cell RSB? It's a single cell reduced representation bisulfite sequencing. So it's one of modification of bisulfite sequencing for single cell data. So also the main goal of doing that is to correlate these methylation island, methylated regions in different promoters and genes transcript abundance by itself. As you remember here, also we can use single cell trial-seq for this analysis. And also, let's mark that this chromatin accessibility, this epigenetics is here, that you can track the epigenetics also on the layer of chromatin accessibility. So here we talked about methylome. And here we will talk like the second group of epigenetics and transcriptomics single cell multi-omics studies is about chromatin accessibility. So one of the methods which might be widely used is related to, is called single cell scRNA-seq. And SciCAR stands from single cell combinatorial indexing and chromatin accessibility and mRNA. So most of these methods are similar, like to attack, but by developing these protocols, scientists became able to analyze this simultaneously with single cell RNA sequencing. So let's discuss briefly other two methods. I will just call them and support you with the presentation when you can read about them a little bit more. So the other two methods are scRNA-seq and SN single cell NMT sequencing. So what is a scRNA-seq about? So scRNA-seq is from single nucleus chromatin accessibility and mRNA expression. So be careful, like single nucleus accessibility. So what you do there? So first of all, you apply microfluidics approach for cell isolation, and then do open chromatin tagmentation using transposase 5, followed by dual omics capture. So you do this profiling of open chromatin regions and single cell transcriptomic studies. So this method allows to observe both these accessible chromatin sites and mRNAs from the same cell. So the important is that, of course, you can apply different omics for the same replicates of your sample, but still tracking these changes on the cell type is very attractive direction of studies. And the last method of this group is single cell NMT, and it stands from single cell nucleosome methylation and transcriptome sequencing. You start with FACS, and then do CPG labeling and bit-based separation of nucleoside cytoplasm, followed by bisulfite treatment. So actually, this method could be even said it like this. Let me rewrite. No, it's not good. So the place of this method is on the border of single-cell methylome sequencing and chromatin accessibility studies. And let's look into this wet lab basis of these tools. What's going on both epigenome and transcriptome sequencing. For single cell bisulfite sequencing, you start with whole cell lysis and just the adaptation of bisulfite sequencing protocol to single cell resolution. So you can do the single cell bisulfite sequencing for DNA that you captured and regular SMART-seq2 or like other RNA sequencing approaches because you applied the bits which used for separation and conjugating your transcripts. What is in the single cell reduced representative bisulfite sequencing? As you can see here, in this approach, you apply both single cell RNA sequencing, in particular SMART-seq2 protocols, and the reduced representative bisulfite sequences scale it down to single cell resolution. In DNA methylation and scRNA protocol, you have this indexed RT primers for RNA, and that is why you know which of transcripts came from RNA, which of DNA, and can distinguish them from this side. And also do the indexes transposition from chromatin. So what you can observe from here is that you split your sample in order to be able to do this both single cell ATAC-seq and single cell RNA-seq. I highly recommend you, if you are interested in one particular protocol, to go deeper into this protocol and look better actually what kind of materials, what kind of optimization do author provide to readers and so on and so forth, because honestly speaking, all of this protocol related stuff is very technical and yeah, and not so widely used in like everyday practice for people in single cell. So usually you specify yourself as a person who work with one particular type of analysis and then follow this analysis. So here I just would like to highlight the other potential protocols which you can use and yeah, and how do they work. The last method in single cell epigenomic studies related to chromatin accessibility is scRNA-seq. It's a single nucleus chromatin accessibility and mRNA expression sequencing. And what you do is, first of all, you extract the suspension of nuclei, then you treat them with transposase in order to receive this open chromatin regions and receive this, to do this open chromatin tagmentation. And then encapsulate all of the cell content into the droplet in order to be able split, in order to be able to target your reads or like molecules of DNA and RNA by different primers and prepare for RT, reverse transcription, and PCR. So yeah, I think the last method and the last set of multi-omics analysis that I would briefly highlight is related to measurement of both transcriptomics and proteomics. And to do this, you might guess that you will apply such instruments or like such reagents such as antibodies. Why to do this with antibodies is that with antibodies, you can conjugate them with different oligonucleotides in order to be able to correspond this particular type of antibody with this oligonucleotide. So actually the idea is to first of all treat your sample with antibodies and do this antibody matching with their target, with like other proteins that you are interested in, and by correlating this protein to antibody and the barcode which antibody handles, you will be able to prepare the sequencing library, and then actually in your sequencing data set, look at this like protein barcodes, and like which corresponds to antibody and like proteins by itself. Of course, because of these multiple steps, these technical bias are quite intense, but still it's very informative and very powerful method in single cell multi-omics analysis. So I will list most of these methods which also use different antibodies approaches. And here you can already guess, the SCC that we talked recently, and use antibodies for targeting self-surface protein. But also this set of methods includes other approaches, such as CRISPR-based approaches, such as scRNA-seq. So in brief, just not to go super deep in these studies, I will just highlight that PIA or STA is the proximity extension assay and specific mRNA targeted amplification. So it's actually the method which was widely used for biomarker discovery, and it's based on these oligonucleotides labeled antibodies, and they are binding to particular protein products. In this method, you do this cell isolation, like using different like mostly microfluidics, to be honest, and prepare the library by this concept that I described before. However, here on this small representation of single cell multi-omics, I would like to briefly interrupt and look and see, like, and discover a little bit other single cell omics and single cell multi-omics tools which were already developed. So as we discussed right now with the single cell, with the couplet measurements of single cell proteins and single cell transcriptomics, you might use this oligonucleotide barcoded antibodies, but also you can do the single cell proteomics, like pure single cell proteomics by itself, and then match this with pure single cell transcriptomics or other single cell multi-omics data. And for calling single cell pure proteomics, I mean really pure proteomics, like based on mass spectrometry and other analytical techniques, but most intensively it's a mass spectrometry. So the single cell proteomics, as far as I know, was very intensively developed in Slavin lab, and this method is oriented on separation of each cells and this their realization of different procedures such as sonication and preparation for mass spectrometry. So the main difference is that with antibody-based approach, the list of proteins that you are going to observe in your data set is restricted by this number of antibodies that you are going to apply. But if you're talking about, and if you would like to have a real high throughput method, like mass spectrometry is much more suitable for these needs, because in mass spectrometry result, you have different peaks of relationship between mass and charge of different molecules or part of molecules, based on which, and using like different databases, you may say that this particular protein is abundant or like very intensively presented in this data set. So single cell proteomics by itself is very perspective direction, and I really love the mass spectrometry based single cell proteomics because it's much more high throughput. However, because the sample of this sample preparation is quite complicated and the amount of biomaterial which you use as a sample is very low, it also promotes a lot of technical bias on this data interpretation or other stages, like literally tracking the peaks and like discovery of false positive results and so on and so forth. So yeah, this method scope on scope 2 is based on this manual selection or like separation cell suspension in plates, 384 well plates, and then preparation this plate content, first of all for liquid chromatography in order to split these different products and like different proteins on this fraction, and consequently mass spectrometry when you look into this relationship between size, like weight, molecular weight, and charge, and identify your target molecules there. So, but single cell proteomics is not the one tool which you can also apply to your use in your single cell studies. And I was really surprised because I have not been thinking about that even such a new discipline as single cell lipidomics arose less than 2 months ago. So it's a preprint from end of the February this year. And of course, we do not have much time to discuss it, and I was going to actually have this interaction with you in order. Maybe you have some suggestion how to do the single cell lipidomics analysis. But let's cheat and say that this method is also used on mass spectrometry. But before you stain your sample, you do the imaging of this. Of your sample, and then on the regions by this image processing, you find these regions of intensity of this of this cell content, where which you after that using laser separated from this sample matrix. So each of the sample after that is proceed with electrospray ionization, liquid chromatography and mass spectrometry, and then you also you can do like similarly to mass spectrometry based proteomics, like in general all of these mass spec methods, you can do the identification of different lipids. What I really love of this paper is that actually I was surprised that this data processing approach was pretty similar to single cell transcriptomics by itself. So after processing of different samples from inserted from these matrices, they proceed this as a list of different molecules and the intensity, like approximate amount of this molecule and in the sample, and did this PCA analysis. So here you can see that some samples are characterized by higher amount of one particular type of lipid and or absence of another. So it's like blue is like negative PCA, and red is high. So I was just like to sum up, I was really surprised that it's very similar to single cell analysis. In here, even if you will look at these pictures without any content, so here, for example, into this single cell data, if you will highlight or like even remove this peak 1, peak 2, peak 3, you will see even the same table for single cell count matrix already with maybe normalized or some processed, like after some processing procedures. And further steps of data analysis is the same, so like normalization, scaling, and so on. Based on this data, they did clustering analysis, and I really love that it looks really similar with the single cell data. And lastly, they validated this lipid content and single cell like transcriptomics, and see that based on this clusters that they observe on lipid layer, they could also correspond this cluster with single cell transcript changes. So here on the figure A, you can see this regular single cell transcriptomics experiments for the fibroblasts that they treated. On the figure B, it's a heat map of different cell clusters and like cell group of clusters with different types of molecules. Yeah. Oh no, not. It's the correspondence of these cell types and subtypes, and this on this third figure, you can see this clustering as received by lipidomics profile. So actually, I was really surprised, and I thought that nothing else can surprise me, but then I found the paper of single cell spatial epigenomics. Like I was really thinking that a lot of drawbacks and limitation of wet lab methods are very crucial. Like you have to put a lot of effort on solving different technical issues. And I was really excited that a lot of people are investing and doing the research on optimizing and developing new techniques. So similarly to single cell transcriptomics that we slightly discover and discuss on the first lecture, you can do as well as single cell epigenomics. And yeah, and also it is a pretty new, like fresh method. What you can do, like instead of single cell transcriptomics, you try to prepare your sample and pick just one particular region and do this manual separation of this assigned cell types and cell clusters to the plate that you are going to proceed after that. So similar to single cell transcriptomics, you do not receive one cell profile, cell specific, but some kind of bulkified representation of the cells with the same proximity. So with the cells which are standing nearby. Yeah, and then you do the data analysis and try to correspond with the cell location. So in your plate, you do have different barcodes which are linked to localization, and then you assign these samples from different locations of this, like physical locations of your sample, and these barcodes which you used in the, for like before library preparation in this plate. And that was one of the most exciting things that I read recently, is a spatial single cell multi-omics. So actually you can also try to do multi-omics with a spatial resolution. I think there is no need to describe why spatial resolution matters, because of course, to do this droplet generation and have a very throughput method, you have to disrupt cell context. And it's very interesting to see actually how literally particular cells behaves and like changes according their microenvironment or like cells nearby. So that is why single cell multi-omics is also a very interesting tool, which was developed less than half a year ago. And this tool is based on, here in this case, measurements of transcriptomics and genomics. And as a sample preparation, you have a slice of your biomaterial. Then you do staining and mark your sample with some fluorescence molecules, in particular in this case DAPI, which is used for highlighting the sites of high content with adenine and cytosine. So in general, you do some kind of immunostainings and immunofluorescence, sorry. Yeah, and then based on this slice, you try to extract both the DNA and RNA. RNA here as well, as I remember, collected by poly-A tails, and then try to do this library preparation for both single cell DNA and single cell RNA sequencing. So in brief, first of all, you do all of this permeabilization, immunofluorescence reactions on the slides, yeah, and reverse transcription in order to be able to receive your mRNA and remove your tissue. So you do not, how to say it, make the tissue suffering on your glass. So you just extract the DNA and RNA from there, and then start this library preparation. So for this protocol, there are two options. So first of all, you do the second stain synthesis and extraction of cDNA using bits, and then you supply it with in vitro transcription, or you can do the RNA ligation, bit purification, and second strain DNA synthesis in this solution that you operate with. After that, you apply, you can go through qPCR to see the amount, look at the amount of transcripts that you have, or like DNA that you have, or do the sequencing using NGS platforms. So I see that we have not so much time left, and I will let's talk in brief about not a wet lab side, but a computational side of multi-omics data integration. So I believe that for people who are there, it's the most tasty part, and but I will try to make it explanatory. So the main task on how to integrate the data that you received from different different single omics experiments with the same sample, or these simultaneous measurements of these omics modalities in the one sample, you faced different tasks, in particular QC, like how to prepare these like such a general tool, general step, how to do the proper filtering and selection of the data that you are going to take into analysis further, how to do the data standardization and normalization, like usually it's possible to do for in these borders of one particular protocol, but but if you are going to operate with different modalities simultaneously, you might also go into it at one moment. And dimensionality reduction. Here is the biggest space for development, because in multi-omics studies, quite a popular and appropriate approach is to do a joint dimensionality reduction. So you have some data for your samples in both in these different modalities, and you try to all of these modalities find the most important and relative information for your sample. So that's how joint dimensionality reduction works. Also, you might do the label transferring. So for example, somehow extrapolate the biomarkers on the protein level and mRNA for the cell clusters. And it's also the question how to, it's based on the joint dimensionality reduction, but still it's the question how to do this proper distribution and assigning of the cell cluster, cell type cluster from different omics. Yeah, and of course it's very important and big challenge to do the result interpretation, because you have to intensively search in the literature to look on the evidence of different observations that you might find in your multi-omics data. So let's discover one particular, a little bit of practice or like example. So imagine you have a sample and you measure transcriptome, copy number variation, like on the DNA level, microRNAs content, and methylome in these cells. What you can do is is the idea, and here is what is presented is you have to transform your raw matrices into do some matrix operation in order to first of all overlap these different markers and types of data for different cell types, and after that assign this to this different metadata and do this grouping and interpretation on the higher level. So I will discuss later on other approach, but if you would like to know about this more, please feel free, and I highly recommend you to read this publication called Integrative Methods for Practical Challenges for Single Cell Multi-omics, where all of these methods and like most widely used methods and approaches are intensively described. So in brief, what do you do with your data is that you start with that you can start overlapping this information if you are sure that these modalities came from the same cell, or you can do this unsupervised clustering and integration using different statistical tools. So here is the pure bioinformatics and like data sciences wakes up and you apply a really huge variety of different protocols to interpret your data. So you can do canonical correlation vectorization, or you can do variational bias, or you can build some linear models, and so on and so forth. So for these different tasks, as you can see, like feature projection, like bias modeling or regression modeling in the composition, you'd use different tools. And also you might have to take into account that all of these tools have their limitations, and some of these tools are more suitable for one particular type of data and not suitable for others. So it's really, really big and complex art to find the proper strategy for data processing, in particular in multi-omics, because single cell data is by itself very sparse, and in single cell multi-omics, all of these challenges are like even much more bright and much more crucial for your data, in your data analysis work. So all of pre-processing of this data is related to quality control, as we discussed, the data normalization and some imputation of the data. For this needs, a lot of tools were developed, such as Seurat, MOFA, Mimosa, Music, and so on and so forth. And as you can see on this column in the middle, they rely on different statistical approaches. That is why they have different functionalities and cannot cover all of these needs, or like they don't have all of these possibilities at the one moment. So some of tools are appropriate for QTL mapping, some of tools can operate with spatial information, and you should really be very precise in your needs and understand which kind of information you are going to proceed and which kind of results would you like to obtain by selecting these particular tools. But most of widely used tools were benchmarked in this paper, and here in the graph right, you see this cell spatial image from this data set, which, from data set published before. And in the figure B, you see the results of benchmarking where these tools, MOFA, Liger, and SRA3, were compared each to another. So as you can see, MOFA, or multi-omics factor analysis, which I will describe a little bit on the next slide, has the highest score of matched data. Processing like matching information, integration of matched and unmatched data, and that is why it is. Oh no, sorry, it's my mistake. MOFA has this biggest score for matched data, but SRA3 has this biggest score for unmatched data. So what means matched and unmatched? It means is data came from the one experiment, from the one experiment. Also, all of this method varies on the functionalities and reproducibility, visualization, all of these advanced features, which are simplifying your interaction with data set. But in general, first of all, they are different on the basis of the approach, like statistical method, which underlines this method. So just to make you familiar a little bit with one of the tools which could be used for multi-omics data integration, it's MOFA, multi-omics factor analysis. And in these tools, you can do unsupervised analysis of the data. So it means you even cannot know if the samples were taken from different, it's better to say that you are not obliged to operate with metadata in this case, and you can already find some results and some insights and changes with unsupervised methods. So you do not have to operate with some preliminary information of your sample. So also, what is the one of advantages is that features that you are going to observe in using MOFA can even be the overlap between these features could be absent. So it means that you can literally see, analyze these different data modalities without no overlap, because usually you do overlap to match these data and do this assigning of different clusters or group, or explanation, like linking different modalities. And here it's good that you can operate with no overlap. And also this analysis explain variability. So what you do in your data with your data set is that you try to decompose the matrix which you have for different like for a set of matrix with assigned weights for these features, and also a set of matrix with latent factors. So there are actually two steps, which you first of all assign different weights for these different biomarkers, or even some association, some factors that you have for your data set. And then you try to find these factors which explain variability, and also let me check. Yeah, I have it. Variance and the content, like the impact of these factors on different groups. So first of all, like MOFA could be presented as a two-step clustering and representation analysis. So first of all, you split your sample into the groups preliminary, and then you try to do this decomposition and understand which of factors in different groups are most explanatory related to variance inside these groups. So the idea is to highlight which of this method explain other factors, and like which of these different biomarkers could be grouped together. Yeah, so in brief, the limitations of multi-omics analysis is that, like, just on this technical side, is that first of all, it's a very big data set, much more than so actually you group everything, and that is why the size of the data set arise, and the power that you have to put to analyze this data set also rise up with non-linear correlation. And also that is why it might cost a lot to handle this data and to process this data. Also, there is a very big need for these integrative tools, because still some kind of golden standard for multi-omics analysis for both wet lab and computational methods was not set. And that is why integrative tools are needed to be developed. And still, there is important to understand this interference between the results for different cell types or different samples, and understand factor co-interference. So it means that, okay, for example, using MOFA, you have observed one factor, but still it's not linking this covariation or like a co-interference with other factors. So maybe this factor is not important, not by itself, but because of persistence with other factors, like by this grouping. I'm finishing. Yeah. So, and lastly, why multi-omics is still so incredibly awesome and exciting field is that it allows you to enhance and make your biomarker discovery much more advanced. Also, this process uncover novel drug targets and some therapeutic mechanism. Also, multi-omics was widely used for discovering developmental processes, and it provides us with understanding on how biological processes are developed, and like what are the circuits of these developmental programs. Why I do study multi-omics and I am very passionate about it is that because I believe it's very directly applicable to personalized and like other P4 medicine. And based on this, you can also do patient stratification, like samples observation in much more advanced level. So I think it's, yeah. Just would like to highlight that we finished with our theoretical part, and I would like to highlight that what was not included and where you might go deeper if you would like to know more is that we have not highlighted CRISPR-based methods, like just slightly with scRNA-seq. And also we did not include intensely spatial transcriptomics, but still we gave an overview about its principle on Monday's lectures and today. And also there is a real need to understand in general both single cell by itself and single cell multi-omics or whatever by observing some showcases like publications or also some of these talks or like presentation of people who did this comprehensive analysis and review. So starting from sample collection, like hypothesis, how to say it, setting the hypothesis or selecting the hypothesis that you are going to test till the data analysis and result interpretation. So sorry, please, for taking extra 10 minutes on the theoretical part, but again, feel free to contact me on my social networks, and I will be glad to answer your questions now and our Slack channel on single cell multi-omics days 3.